I'm kicking this off. Paola already introduced us. I'm happy also about, I would say, the collaborative effort between the education and the standardization and open science committee, which Miriam and I are co-chairing. So we are going to guide you through BIDS, and so this is a data structure. We try to start a bit with a motivation, why all of this might be relevant to you. Then Miriam is going to do a very brief overview of the SNIRF standard and the BIDS standard. And I'm going to very briefly dive into Cedalion, which is the toolbox on top of which we have built the tool that is now basically available to everyone. And Shakiba, who has done really most of the implementation, is then going to do a hands-on demo to show how to use this tool. Just to say that that tool is not really needing you to do any analysis in Cedalion. It is mostly just using kind of background features. And so you could just convert your data and use that data in any other toolbox. Okay, and in the end, as already was said, we have hopefully some time for question and answers. So why do we care about data sharing and standardization? Recently, together with 38 labs internationally, a team which included Miriam and I and Rix Mosquita and Rob Luke did what we call the FRESH study. And the FRESH study asked 38 labs, people from all over the world, to analyze the same fNIRS data and report what results they found. And these results were very different. There are very interesting consistencies. If you're interested, you can read the paper. But what's also kind of course, fun to see is that the path to the result has been very different for all participants. What this leads to is that, of course, it is not always easy or usually very hard to reproduce results. And this led to the so-called reproducibility crisis in fMRI and EEG, and fNIRS is facing really the same challenge. So how can we make sure that everybody across the world, when they analyze the same data and use the same analysis framework, kind of end up with the same results? So that would be where we would like to go. And another thing that is very useful, of course, from a point of machine learning, which my lab is amongst others heavily involved in. If we have standardized data formats, we can learn much better from a variety of data sets. So what is important for this? First of all, a standard for the data itself, which is in our case the SNIRF standard. Miriam is going to briefly introduce this. If you want to see the little paper that introduced the standard, here is the citation. Then the data itself has to be structured in a certain way and provided alongside conventions regarding names of folders of data, experimental information, and so on. And so this is across different modalities, the brain imaging data structure, which now is also available in NIRS. And this is why we're doing this webinar as well. So if you want to read the kind of introduction into that, here's another paper reference. All of this doesn't really help if the data is not findable and accessible. So that's another part which is kind of linked, and we'll briefly come to this. So, platforms so you can upload your data. And again, ideally, then the uploaded data follows the standards. All of this is a global effort among many different labs and institutions. One of the efforts that we are doing in the standardization and open science committee at the fNIRS is trying to make this more transparent. And so if you want to learn more about this, please go onto fNIRS.org. We also have a newsletter there now that we have advertised. And I see session expired. I hope I'm still there for everybody. Okay. Please call me if I'm suddenly gone. Okay. So if you are interested in this newsletter, if you're interested in standardization in general, then please go there and sign up. Okay, so what's the vision? The vision, as I already mentioned, is ideally at some point in the not too distant future, anybody could reproduce your results anywhere. What do we need for this? We already mentioned the data has to be available. So there's cloud platforms. There's, of course, the platforms like Nitric and so on that in the past. There's OpenNeuro now. And there's services, you could say, like NeuroJSON from Jinjin Fang. The idea is that if you provide your data on one of these platforms in the right standardized format, which is BIDS, Then if you have a tool that can run an analysis, and this could be Cedalion, can be anything else, that also is cloud compatible, then kind of one of our ideas or visions is that with a single click, you could run a notebook, for example, from Cedalion that runs on Google Colab, that automatically downloads your open data that you have stored, for example, on OpenNeuro, and that gives you the results. And anybody could run that anywhere and could also change parameters in pipeline to see how the results change. And again, of course, the scales to machine learning, which is kind of a personal motivation for our lab and others. So why do you care? Aside from what we already talked about, of course, it increases impact, visibility, and credibility of the publications and the data that come out of your work. And yeah, we mentioned already, so there is quite some momentum. And I think one of the nice things is showing we're happy that we can do this webinar, and we're going to get hands-on now. We want to say thanks to the creators and the maintainers and adopters of these standards, so SNIRF and BIDS maintainers and creators. Then, of course, there's platforms, and it's not the only one, but like the fNIRS platform, where there's basically open source, which work for software and hardware. The idea is also, of course, that this allows people to reproduce. And the kind of last important pointer in our own interest here, there is going to be a special issue in SBI Neurophotonics called fNIRS Commons, Datasets and Tools for Open Reproducible Science, which is edited by Chin Feng, Miriam, Adam Eggebrecht, and myself. And the idea is, of course, that if you have datasets that you are, after this webinar, able to put into the BIDS structure and you upload to one of these platforms, then we will invite you to submit this data alongside dataset descriptor or paper to this special issue to make this data available. And with that, I'm handing over to Miriam to give a bit of an introduction that goes a bit more into detail to teach you about SNIRF and BIDS.
Alex, you were gonna stop sharing, or?
It looks like I did. Yeah. No, I didn't. Anyways, no, I did.
Okay, now it's good. Okay. People can see my slides?
Yes. Yes.
Yes?
Yes.
Okay. So in the next 10 or less minutes, I'll go over SNIRF and BIDS, just still giving a little bit more detail, but not too much detail because of the time limitations. So shared infrared spectroscopy format. Actually, this is, as far as I recall, first discussed in SFNIRS conference in Japan with the leaders, and the need was there, and then the SFNIRS committee put together these leaders, experts in the field to create this format. This is an open standard for storing and sharing fNIRS data. It's in HDF5 format. It is hierarchical, efficient, widely supported. It's already published. There's a paper explaining, Alex already cited that. So a bit more detail on what we mean by SNIRF. So SNIRF has it's an object, it's a class, and it has different fields. The main fields are format version, metadata tags, data, stem, probe, aux. So you need to have these in your SNIRF class. And each of them has their own subfields. Some of them are required, some of them are not. For instance, in data, you have data time series, time, measurement list, which also has its own subfields. And they match. For instance, if you have one data time series, the first data time series match the first entries of the measurement list. So you can figure out the source detector pair, the channel that corresponds to the first data time series, etc., etc. So quickly moving to the brain imaging data structure, this is a community-driven standard for organizing neuroimaging data. The need was there in all neuroimaging fields, not just us. And it was coming from the fact that many labs, or every lab has their own data organization. So it was hard to share data or use the same code, for instance, with different data sets. So you need to reformat the data, etc. So this has been kind of solved by this BIDS structure that was introduced. It's designed to support sharing, validation, and automation of datasets, and it's widely adapted for fMRI, EEG, MEG, PET, IEEG, etc., and other modalities. And it's supported by tools, pipelines, and repositories. And this was also, again, introduced to our field because we also needed it. And there was this effort for extension of BIDS for fNIRS, led by Rob Luke and Luca Polonini and others. It's also published in Science Data this year. So now it extends the BIDS structure to NIRS data, continuous wave, time domain, frequency domain, and DCS. And it builds on top of SNIRF to organize full data sets. So you first have to have your data set in like this individual data files to be in SNIRF format. Then you can put all of your data set in BIDS structure. What do we mean by BIDS structure? So it defines a folder structure. So it's predefined where you need to put the subject files, session files, etc., everything. Metadata files, everything's location is predefined. File naming conventions are predefined. For instance, you can see here, you need to put the subject ID, the task name, and if you have multiple runs, you also need to have the run, for instance, run 1, run 2, etc., in the file name. So the file names are also predefined, so you need to follow that. And there are also standard metadata files. For instance, there's participants TSV file. So if you want to include, for instance, for all your subjects, the age, sex, and like other demographics or like patient groups, etc., you can put all of them. And this file is in the parent data folder you can see on the left. And there are also events TSV files which explains the timing of each like stimulus, as well as like the name of the condition name, for instance. And there is also data set description. That's kind of a summary of the whole data set. So combining the two, why we're doing all of this, Alex already motivated the whole thing. So of course, we do all of these to be able to easily share our data sets. So that, of course, reproducibility, increase reproducibility, but also this will allow other labs to use our data sets easily and, you know, create different results and increase the benefit from the same data set. How we can share, once we have the our data in BIDS format, we can share it through OpenNeuro. This is a public neuroimaging repository, and it already accepts SNIRF and BIDS. And it's free and maintained by the community with an NIH support. And actually, Shakiba, at the end of her demo, will also show us how to, once you have your data in BIDS, how easily you can upload it to OpenNeuro and publish there for everyone to access. And another effort that I want to highlight is the NeuroJSON platform developed by Chin Fang from our field. This is the next generation imaging data hosting and search engine, so it's also worth to mention here. And lastly, I just want to highlight that there are validators for both SNIRF and BIDS. For SNIRF, you can search GitHub and PySNIRF2. That's a Python-based SNIRF validator, and it also allows you to, of course, read and write. And there's also a BIDS validator. Again, you can search GitHub BIDS validator. Or, as I said, OpenNeuro already has a validator. So once you upload it, it will already take your data through the validator and tell you if there are any like warnings and errors, etc., and report that. And actually does not allow you to publish it until you fix the errors. So that's pretty much it. I'm stopping. Okay. I'm also muted. You're muted.
Yes, well, I was saying hopefully smooth transition with that. Of course, I messed that up. So you should pretty much see the same as before, but on my screen, if I forward, I see a nod. Yes, okay. So I'm going to briefly kind of talk about Cedalion, which is the toolbox that we're developing at this lab. It's only like has a peripheral role here, but since the tool that you're going to see displayed by Shakiba is building on top of Cedalion, I want to briefly motivate it. So we are working on fNIRS and diffuse optical tomography time series. And the whole toolbox is motivated by us trying to go into the everyday world with fNIRS and DOT. And we have a lot of problems, as we all know, in the community with measurement artifacts, but also with physiological confounders. And as soon as we go into everyday world, then we also are often not really knowledgeable about the labels and stimulus conditions. So the idea is that we include other modalities, other physiological sensors, for example, ECG, eye tracking, respiration, and that, at least in our lab, we are using machine learning for sensor fusion to explain variance in the data, to improve the separation between brain activity and confounders. And so this is funded by an ESC starting grant, which is therefore also funding quite a lot of the Cedalion toolbox development. So the idea of this toolbox is that it kind of combines advances in the field, which is, first of all, machine learning, as I already mentioned, but also diffuse optical tomography is making progress both in the methodology and modeling, but also, of course, in the availability of hardware, of instruments. And the toolbox is, its lead architect is Ike Medell, you see here on the top. What it entails, just in case you're curious, for the future, it can do a state-of-the-art pre-processing and analysis that you know from fNIRS, from the fNIRS domain. It includes different kinds of head models and atlases, including individual, but also standard atlases. It allows you to work in parcel space and networks. By including toolboxes like MCX or NearFaster, it allows you to do photon simulation and then allows you to do imagery construction if you care about DOT. We have a few features I want to briefly come back to in 2 slides that allow you to add synthetic ground truth activation to your real data to have a ground truth for methods evaluation. And of course, because it's in the Python ecosystem, you have pretty much all the things out there like scikit-learn, PyTorch, and so on to directly interface with your fNIRS data. And all of this is only, let's say, hopefully valuable because it builds on top of the giants, of the pillars, which are the standards, which are the other community toolboxes from the fNIRS community, but also other fields like MNE and EEG and others. And of course, big thanks to all the contributors in the community so far. So just to give you some images, if you use it, what you can do is you can use your MRI, create an individual head model, use photogrammetry for optode co-registration, do forward modeling and photon simulation, do imagery construction, use my brain during finger tapping. And now, since we're already in the right toolbox, we can also do machine learning, single try analysis, classification. Okay, but that's not really so relevant for the webinar. Just a last shout out that this new release in summer gives us a few new features, which I already mentioned: Schaefer parcellation, synthetic augmentation, multimodal decomposition methods for fNIRS and EEG analysis, and a few other things. Much more relevant for the webinar, and this is kind of the short loop I wanted to do is, of course, the toolbox is open source and freely available. You can download it, you can run it on the cloud, and if you want to do so, first of all, to get access to all the resources, you can go to cedalion.tools, and you will get this collection of images that bring you to documentation, GitHub, etc. For the webinar now, as you might have seen already in the instructions that Shakiba sent around, the example notebooks that we have in our documentation and also in the toolbox code itself have an open in Colab link. So you can click on that and it will open the example notebook in Colab, which is Google's kind of collaborative cloud-based Python environment that can allow you to run code on the cloud. And so this is exactly what we will see now also on the notebooks that, or on the notebook that Shakiba has prepared and will guide you through now. So if you want to follow her hands-on, then ideally you use the link and open it in Colab. I hope, if you wanted to do that, you might have already tried it out. But this is where I hand over to Shakiba to actually get concrete for what I think most people came here for actually, and that is how to use this tool to create BIDS compliant datasets.
Am I sharing this? Am I sharing my screen?
Yes, maybe you want to put in presentation mode.
Yes,
Yeah,
okay. Okay, hi everybody, I'm Shakira and I'm going to tell you about how you can make your datasets arranged like BIDS structure using Cedalion. So we will first have an overview of the main steps we should do in Cedalion to go for BIDS format. And then first we will navigate to the Cedalion documentation to find the example notebook we provided for this purpose. And then we will try it with probably one sample data set, and then we will have a like collaborative session with the shared sample data set. We shared it with you before. In this section, you can also ask your questions in the chat box. You can try it out simultaneously, and then we are going to validate the output using the OpenNeuro validator. So in Cedalion, for creating BIDS structure, there are multiple steps. And here there is an overview over the steps we are going to take. The first one is creating a semi-automatic mapping file for extracting as much as information you need from the SNIRF files. And then we are going to check for any scan or session information from the SNIRF files. And we will see them in the notebook. And then we are going to rename the SNIRF files according to the BIDS naming convention, and we will create a BIDS structure directory to move SNIRF files to that directories. Next, we are going to generate all modality specific files, and then we are going to generate modality agnostic files. And after that, we can just modify our files according to according to the things we want to add to the files at the end. So if we just move to the Cedalion Cedalion documentation, we can see that there is a data structures in IO section here. And if we go into that section, we can see examples, and this is the example notebook we are going to work with today, convert the fNIRS dataset to BIDS. And here is the button Alex explained about. If we click on it, we can just open it inside Colab environment. So here in Colab, if I just run the first cell, I can see that it will just connect the session to my Google Drive. I should just authorize it. So previously, I have uploaded my datasets in my Google Drive. Here you can see 3 datasets, and now I will see the datasets in in the Colab as well. So it says that it has already mounted my drive. Here I have a drive folder. Under my drive, I can see the webinar datasets, and I have 3 datasets. I'm going to explain about one of them or 2. It's also installing Cedalion inside Colab, so you don't need to do anything locally, but it takes some time. So yes, then you will see this red warning, but don't worry, it's okay. The session is restarting and we can just move forward. So if I just run the first cell, you can see that Cedalion is already installed in Colab. I have also the dataset folders here. So there is a demo mode in Cedalion, which I set it to false. If you want to try it, you can just set it to true. This demo mode means that you can easily get the sample dataset from Cedalion cloud. But now that we have our own dataset, we will work with the demo mode set to off. So here I should just define the dataset path. I will try the second dataset from Artemis. I will copy the path here. And now I just need to define a destination folder. I will use the same name, but with BIDS. Sorry. So here we can just add any kind of metadata. I mean, this is optional, but the field that you are adding here, it could be just a JSON file or directly go to your data description file. But now it is optional and we don't use it. And then here we are trying to create that mapping file I explained about. So now we can see that under the folder, under the dataset folder, we have an extra file called SNIRF to BIDS mapping CSV, which we're going to modify it in a few minutes. So here, if we have any kind of participants CSV file or JSON file, we can provide them, but since they are optional, I mean, I will let the notebook to create them itself. Now, if we look at our SNIRF to BIDS mapping CSV file, we can see that we have multiple columns, but all of them are none, which means that the function couldn't extract any information about subject name, session, or task from the SNIRF file name, but we should provide them. So I'm going to download this CSV file. And here I'm going to modify it. So the subject name here is KD, I assume. And the task is, I don't know, you can just, I will just use vision simply. And could be any kind of name you put for your task name. And then we can see that there is 0, 3, 1, but this is, I mean, the format of this of the column is numbers, so we need to change it to text so that we can put 0 before 3, 0, 1, and 0, 2. And so for BIDS structure, it is essential to have subjects and tasks, but all other columns are optional, like acquisition, run, and session. But then, I mean, you can just modify it as much as you need. But here we have also condition, match, and duration. So these are for event CSV file, and I will explain it in, I will explain these columns in the next dataset I will show you. So now I will just save it like it is. And here I will upload it again into the folder. And if we open it again, we can see that the subject and task and run are filled out. So in the next step, it is going to search for any kind of information about scan information, like if there is any scan.tsv file provided in the dataset folder, which we can see that there's nothing provided here, or it can extract scan acquisition times from the SNIRF file itself. So if I just run this section, I can see that at the end, there is a new column here called acquisition time, which is related to the scan acquisition time, and it is extracted from the SNIRF file itself. And so we are going to do the same for session.tsv, but if I just run this cell, nothing happens because, because we don't have sessions in this dataset, right? So session acquisition time would be just none. So now it's time to create our BIDS folder structure. And for that, we need to have another column, which is BIDS name. And these BIDS names are created according to the information we had in our mapping CSV. So for each scan file, we are creating some BIDS name, which are following the BIDS naming convention. And then we are also creating some parent path to know that we are going to move this SNIRF file to this parent path in the destination folder. So now we have already the information about the tree of BIDS folder, and we are going to move our file and renaming them according to BIDS structure. So if we just run the next cell, we can see that there is another folder here, dataset RT-NIRS BIDS. If I open it, I can see semi-BIDS structure. We have subject level here and then NIRS, and then we can see the renamed and moved SNIRF files here, according to the information we filled out in the mapping CSV file. So now I will move forward, and if I just run the next cell, I can see that the modality specific files, like the event files and optodes.json or any other information which are modality specific, are created here. So now if we go forward, we can also create our scanned TSV files according to the information we extracted before. And if I run this cell, I can see that like for this specific subject, I will have a scanned TSV file with the acquisition time I extracted before. We can do the same with the session TSV file, but since we don't have any session, it doesn't do anything. And now it's the time for creating modality agnostic files like participants TSV, participants JSON. So here, if we have provided participant TSV and JSON file here, it could generate those files again inside the BIDS directory. But since we didn't define them, it will go through the either the default participants TSV file, or we can also define a list here so that instead of a species, age, sex, and handedness, it uses gender and age as fields inside the participants CSV and JSON file. So if I just run this cell, it says no valid participants TSV file found, which is correct because we didn't provide any. But then if we come here, we can see that participants TSV file is generated with the fields we passed as input, gender and age. And now if I move forward, we can see that there's another section for creating data description file. And we will see, the only information it gets from your folder is just the folder name, and it will fill out the other information with some default values. But if you have provided extra metadata before, those information will be added to the data description.json file as well. Now here we just fix a field in event in the court system.json file. You can just read about it after the webinar. It's nothing important. The thing is that in BIDS structure, we can't have empty strings, and now it just checks if something is with empty strings, and it will replace it with other. And now we are going to fix the events.tsv files, and first we're going to order the rows according to the onset time, and then we will talk about this in the next data set. But since we didn't fill out the count and count map in the mapping CSV file, there's nothing to edit here, and finally, we can just decide that we want to keep the raw data set we have as a separate folder under the BIDS structure. So if we just run this, we can see that in our BIDS folder, we have now, oh, it's not there yet. Yes, we have now source data. folder, which contained all the raw data we had for the data set. Now we can see the directory, the whole directory of the BIDS directory. So we can see that we have subject level, and then we have modality specific files, and then we have also modality agnostic files, and also a source data directory, which contains the whole data set we had, the parent data set we have. And if we move forward, we can just observe participants.tsv file. We didn't fill out anything here, so they're all non values. We have also participants.json file with the fields we defined before. And also this is our data description file. And the only filled out form is the name of the data set, but all other stuff are just default values. You need to modify them yourself. So now we can see that the dataset is completely in BIDS format now. I want to go for the shared dataset, the sample we have shared with you before. And then we will, at the end, we will just try to upload it into the OpenNeuro and see if actually it is validated by OpenNeuro as well. So now you can also try out with me the shared dataset, which is this dataset NIRS from NIRS. So I will try to do it from scratch so that you can also take the steps with me. So again, let me just delete my runtime. So here we have Cedalion documentation again. We can go to the data structures and IO examples, and then we have this example notebook convert a fNIRS dataset to BIDS. We can just open it inside Colab by this button. And we have now Colab in our, Colab environment. And if we run the first cell, it will connect our Colab to the Google Drive in which we have uploaded our dataset. So now for trying out this notebook example together, if you have any kind of question, you can just write it in the chat box and I will try to answer. So now we have our drive mounted here. We can see the data set. I will go with the NIRS data set. That can take a bit time for installing Cedalion. We just set this demo mode to false because we are going to use our own data set. Okay, the session is restarting and now we have Cedalion installed. Okay, and now we need to define the dataset path, which is the path of my drive, the dataset I uploaded in my drive, or the shared dataset you have. And now there is also destination path, which is the path you want to save your BIDS folder. Oh, again, I forgot to run this up. So here we are not going to add any extra metadata again, but we're going to create the mapping CSV file, and because the mapping so if we also don't have any participants CSV or JSON file inside our dataset folder. So I will just let the notebook to create them. And here, if we just read our CSV file, we can see that again we have non-values here, which we should modify them. So I will download this mapping CSV file, and here our subject is probably 473 and 474, and the task is ball squeezing, I assume. And here we are also going to use condition and condition match, because inside the events, because events TSV files are created according to the data extracted from the SNIRF files. So they could have different trial type values. And I know that in this data set, we have 2 trials, which are saved as 2 or 3 in the SNIRF file. But since we want them to have more meaningful trial types, we can just map them to right and left. And I will show you that this mapping actually can modify the events TSV file at the end. So I will just save this mapping CSV file. You can also do the same. And then we will upload it again into the folder. And if I reload it, I can see that the modified version is now on the Colab. So now we have 2 subjects. We have a task defined. We have also condition and condition match. If we move forward, we can also look for any kind of information about the scan TSV files. So we know that there is no scan TSV files inside the dataset itself, but it can also extract some information like acquisition time from the SNIRF files. And it can do the same with session files, but for session files, again, we have non-values because we don't have any defined sessions. And then if we go further, we can create the BIDS name according to the data, according to the column we provided with mapping CSV file. And here we can just we can see that there should be an extra folder NIRS BIDS. And if you open it, you can see the BIDS structure folders following the naming convention. And now we are going to create the modality specific files, and I will show you the events TSV files after this section. And now we have modality specific files, and if I just open one of this events TSV files, we can see that the trial types are 3, 2, but we want them to be left or right? So we will see that we can also modify it. Here we are going to create a scan files and again session files. Now I would prefer to go with the default values, species, age, sex, and handedness, and then we can move forward and we can create data description file as we did. And this is a fix for coordinate system.JSON file. And now we can order the events.tsv file according to the onset time. And here we should see that having defined count and count match, we have actually modified the events TSV file. So now for trial type, instead of numbers 2 or 3, we have right, left, which is actually, maybe it's more convenient, but it is also optional. you are not obliged to do this part. And then we are going to save this directory to and the raw data folder inside the BIDS data folder. And after this part, we are going to inspect our new data folder, which is we have subject levels, modality specific files and modality agnostic files. And then we have also source data with all kind of data set provided at first. And if we just display the participants file, we can see that these are like default values, default fields for participants DSV. And we have also the JSON file, which is filled with default values again, and also a description and dataset description with the appropriate name and also default values you need to modify yourself. So now I will try to download this new folder of dataset. Sorry. It take a bit time. So here we have the dataset folder, and still we need to fill out the participants DSV and participants JSON file. I don't know, we can just fill out this with some value. Save them, and then we have participants.json file, which is filled out with the default values, right? So I don't need to modify it. And then if I go to the OpenNeuro, you can just easily create your account. And there is an option here for uploading your data set. And the second step is BIDS validation. So if I just upload my data set, I can see that it is validated. So I don't see any kind of error here. These warnings are ignorable. You can easily go further and just upload your data set in the OpenNeuro. So that was the flow. And now I want to say that we will have a Q&A session. You can ask your question, but we also have this forum here. I can again show you in Open fNIRS. You can ask your question here, and we will try to frequently answer them. So yes, and finally, I want to thank people who actually helped this for this webinar. IK and Tricons, special thanks to them, and also the IBS and Boston labs. I don't know, maybe Alex can better say thank you.
I think you did a great job. Actually, what I need to do still, I'm going to snatch away your screen, is to thank Shakiba. So Shakiba did really, I would say, most, if not all, of the hard work on the implementation that you just saw. And so all the credit to Shakiba. And of course, now also thanks to Miriam for supervision and help. So with all of that, and again, credit to the teams involved in some ways or another, we have, it seems like 7 minutes for Q&A, if there are any questions. So let's see if there are any.
If you have any question, you can either raise your hand or you can type it in the chat. Yeah, we have a question regarding the presentation, if you're willing to share the presentation with the attendees, or if you can, I don't know, upload it on the Cedalion website or something.
Yeah, I think there's no problem sharing it, and maybe you can say something about where the video could be found, because I think then you
we
will
will
also get
share the
info.
video, of course, on the
Yeah.
fNIRS website. So I will send an email when it happens. But
yeah, so we'll, I guess what we'll do then is we'll share a PDF version of this through you to the participants.
That's great. Does anyone have any other questions? Well, first, thanks so much for the amazing presentation. I forgot to say that. It was really, really informative, especially for me as well, that I'm not too familiar with this yet. But yeah, this is super helpful to get this started and implemented in our labs as well. So thank you very much for the resources that you have created. Can maybe ask maybe a quick question that might be, I don't know, silly, I don't know. Are there any plans for handling multimodal data? So often, like we record fNIRS alongside EEG, and there is BIDS format for EEG and other modalities. Is this something that can be handled or there will be an opportunity to handle this?
So I think there's multiple layers to this answer. Maybe I give the high level answer. So one thing is, of course, reading multimodal data depends on the toolbox. And there I can say that we want to, like Cedalion is trying to be multimodal. So the idea is that at least the main modalities we're working with, we also want to be able to load from BIDS. So far, this is constrained to basically all the multimodal data that is part of the auxiliary field in SNIRF. So if you have any, let's say, maybe PPG, respiration, or things that you would kind of combine with fNIRS, usually they would, if they are part of the SNIRF file, that would be easily loaded and can be worked with. The other thing that Shakiba and Mary mentioned, of course, is that thanks to BIDS, across modalities, the structure is standardized. And so at least where to find session information, task information, and so on, right? Kind of to align data is basically inherent in the data structure itself. So this is also why I think we're really happy that it gets slowly but surely easier and easier to put data into BIDS. Because let's say what Shakiba has done, for example, she's worked on fMRI and DOT data, and if both of them are in BIDS, then basically the workflow can work with both of them similarly as soon as the data can be loaded. Yeah, I'm not sure if I forgot something to mention.
Yeah, I just want to add, you have already seen all the like fNIRS related files, they go under NIRS folder under subject. So subject folder can easily have EEG, motion, etc. Maybe in the future where there's more need, we can add that to that feature to this pipeline. Currently it doesn't have, obviously, but
yeah, that's what I thought, like having modality specific folders. Yeah, that makes sense. Thank you.
Maybe one more thing to add, kind of as a personal note, because I can say at least when we started, for me, it was not very intuitive. Like what is actually in the SNIRF files, how to get to it, and the same with BIDS. And of course, you can read the specifications, but I mean, most people don't enjoy doing that, and it's not necessarily intuitive. I think it's just worth pointing out that there are more and more tools that kind of spare you that. And of course, the point of this webinar and Shakiba's work here was to basically spare you that. So you can basically try out and understand what the BIDS structure even means by taking your SNIRF files and trying to get it into BIDS structure, and then you have a much better intuition. And I mean, Miriam showed the folder structure briefly, right? And in the end, we have seen it's a mix of conventions of how things are named and where they are stored and what files should be there. But I think similar to BIDS and SNIRF, both of them, if you have any tool or toolbox that can work with them, it gets much easier to understand what it even means and to get things into the right format. So I think learning by doing, I guess, is the idea.
Yeah, I think having sample data and sample files, I think it's super important to understand what you actually need for your own data. So yeah, that's definitely.
Yeah, and just to remind everyone that we will have that active forum. Once you have your data set, probably people have different questions, more questions than now. Feel free to ask there.
Yeah, but I guess nowadays most of all the NIRS devices can be saved in SNIRF file. So I think that's a very good starting point. Does anyone have any other questions? Feel free to type it in the chat or maybe to unmute and you can ask it yourself.
Otherwise, the interpretation is that everything was so clear that there's no questions remaining.
I'm pretty sure there will be questions.
Exactly that.
Yeah, of course. Yeah.
Now it all looks amazing and super
Yeah.
Yeah. So I think then just want to take the chance just again, thanks to everyone involved. So really, it might look easy to run all of this. And so again, you know, thank you, Shakiba. All of this has a lot of stuff in the background that now that it runs, it is easy to run, but it is has been quite some work. So to both groups and everybody involved. Thanks.